All right. Well, first of all, I want to really thank the organizers for giving me the chance to speak to you guys today. This is a really cool meeting, so I'm just really excited here. And so first of all, I want to acknowledge everybody that's going to be involved in the work today. I want to thank everybody in my lab that's had a part in many of the experiments that we are that I'm going to show. I'm going to get my laser pointer on here. Those highlighted are going to have, there's going to be data directly from their work. Some of this work I'm going to show is from the University of Iowa, so I have to thank Dr. Stanley Perlman for his guidance in the work, and also Matt Grunwald, who did many of the experiments that I'm going to show today. I want to thank the KU core facilities, really helped us out with a lot of things that I'm going to show you today. All of our collaborators over the years, most notably Mike Cohen and Charlie Brenner, are two collaborators we've just started with, and I'm going to show some of the data from our new collaboration with them. And then finally, our funding from the NIH, University of Kansas, and also Acknowledge Chromadex for providing some materials for some of these experiments. So like I said, or like the title said, we study a battlefield, and that battlefield is on ADP-ribosylation, all right? So if you're unfamiliar with it, ADP-ribosylation is a common post-translational modification where you attach this molecule of ADP-ribose basically to a protein, either as a single unit, or if you can, you can attach them as chains, and that's poly-ADP-ribosylation. This modification has a variety of effects on protein structure and function. It's important for cellular stress responses, and it's mediated by enzymes called PARPs, of which there are 17 known human PARPs, and they use NAD as their substrate, and I've kind of highlighted the main players in this reaction. Importantly, in terms of viruses, many of these mono-ADP-ribosylating PARPs are actually interferon-stimulated genes and play roles in various virus infections. Finally, this modification is reversible by a number of enzymes, including what we're going to talk about today, which are macrodomains. And here's just a cartoon diagram of this process. You have a protein that's targeted by these PARPs. The PARPs use NAD as their substrate, which put an ADP-ribose molecule on a protein, which is then reversed by the macrodomain. So the coronaviruses, all coronaviruses have a macrodomain, and it's always located at the N-terminus of the non-structural protein 3 protein as a small domain of about 20 kilodaltons. Importantly, coronaviruses are not the only viruses that have this domain. It's also present in alpha viruses, rubella viruses, and hepatitis E virus. Okay, so indicating that maybe it's doing something to enhance the replication or pathogenesis of specifically of positive strand RNA viruses. So you're probably wondering what exactly we mean by a macrodomain. Well, this has really been described as a structurally. So there's a well-known structure of about 20 kilodaltons for these proteins with several central beta sheets that are flanked by alpha helices. And here on the right, you see the SARS-2 macrodomain structure that we solved in collaboration with the Protein Structure Lab here at KU. And if you kind of see in here, you can see these central beta sheets surrounded by these alpha helices, which forms like this, helix, alpha, beta, alpha, helix sandwich domain. These proteins bind to mono-ADP-ribose and their derivatives. And over here, you can see a depiction of the substrate binding pocket of the SARS-2 macrodomain. And of course, they remove ADP-ribose from their protein substrates, as I just discussed. So, and here's a cartoon diagram of how that occurs. Now, the coronavirus macrodomains likely only remove ADP-ribose from acidic residues. While ADP-ribose can really be attached to proteins at multiple residues, including serines, cysteines, and even lysines. And so we think that while the macrodomain can remove ADP-ribose from some proteins, it can still bind to those ADP-ribose proteins and maybe affect their function even if they can't fully remove that modification. Because of this upregulation, or because there are PARPs here that are interferon stimulated, it's been proposed by our group and others that viral macrodomains are there to really counteract antiviral ADP-ribosylation. All right. So to really start, when we started studying this protein, the main thing we did was we used reverse genetics to determine the role of this protein, and starting off in vivo. So the mutation that we really started off with and made throughout many different coronaviruses is this asparagine residue. We changed it to an alanine residue. This asparagine is completely conserved among all enzymatically active macrodomains, even beyond coronaviruses. And you can see here, it is structurally providing hydrogen bonds at that terminal ribose. And we've mutated this in vitro and shown that you mutate this to an alanine, you basically lose all enzyme activity here. So what was quite remarkable is that when we started, making these recombinant viruses, we found that they were basically completely attenuated in mice. And so here you have data from MHV, SARS coronavirus, and MERS coronavirus in all really well-established animal models. And you can see that at a lethal dose of virus, where the wild-type virus is killing the vast majority of these mice, you can see that our mutant viruses have no lethality, virtually no lethality. They have very little, any site of disease. So that's really, I've always found remarkable with one single mutation, we've had that dramatic difference in disease. So that really indicates, that this is a prominent virulence factor for the coronaviruses and could be a potential therapeutic target. So I've spent the last 5 to 10 years studying this protein. And so today I'm going to give you 3 short stories on what we found out about this protein. First, we're going to start with the biochemistry of the macrodomain. Then we're going to switch to talking about the PARP enzymes that I talked about earlier, the host enzymes that are involved in this battle. And finally discuss, the impact of NAD, that substrate, on how that differs, affects this battle. All right, so again, starting with the biochemistry of the macrodomain, I'm going to kind of bring you back to what I said earlier. And so that this domain can both bind to and hydrolyze ADP-ribose from a protein. So again, for binding, you can see that it probably binds to ADP-ribose from proteins that are attached at serines, arginines, or cysteines, or other residues that are not acidic, basically. So we've developed several assays to actually test and measure this ADP-ribose binding. So we have an isothermal calorimetry assay, which actually just measures its binding to a single ADP-ribose molecule. But recently, we've actually developed alpha screen assays and fluorescence polarization assays with a non-cleavable ADP-ribosylated peptide. And so those are actually, we've developed, and they're really robust assays for measuring ADP-ribose binding to an ADP-ribosylated peptide. So, and then secondly, we know that the macrodomain can actually remove ADP-ribose from acidic residues, or from if the ADP-ribose is attached to acidic residues. And there are actually distinct antibodies that can bind and detect mono-ADP-ribosylated proteins. So to measure this activity, we use Western blots and ELISA assays. All right, so now in collaboration again with the KU Infectious Disease Assay Development Core and Anu Roy there, who leads that core facility, we've developed this ADP-ribose binding assay I've just described. I'm going to talk about the alpha-lysa assay we've just developed and show that data with the SARS-2 enzyme. And so you can see here that when you have that interaction, or in this assay, you can see that small amounts of that peptide incubated with quite small amounts of the macrodomain elicits a very strong signal. And we know this is very specific because we can use this with, do this assay with a non-ADP-ribosylated peptide, and we get no binding, very, very little binding. And so this is highly specific, and we find that there's a KD value of about 90 nanomolar for this interaction. So it seems to be very tight binding. And then in our lab, a very talented postdoc named Youssef Al-Ahmad has been developing our ADP-ribosyl hydrolyze assay. So what we do is we take PARP-10, a catalytic domain of PARP-10, and in vitro, when you incubate it with NAD, it will auto-ADP-ribosylate itself. And so then you can actually detect that with that antibody I just described. And then we incubate it with the macrodomain, and we should see a loss of signal. And that's actually what we see here. This is the SARS-2 macrodomain. You can see a significant diminishing of that signal, even within the first few minutes of this reaction. So the SARS-2 enzyme is a very good mono-ADP-ribosyl hydrolase. Now, we've been working on these assays well before SARS-2 emerged. And so we had some other coronavirus macrodomains in our pocket. And so we decided to compare them to the SARS-2 macrodomain. So these are the ADP-ribose binding assays. We have the isothermal telemetry, alpha screen assay, and fluorescence polarization. And I'm not going to go into this too much detail, but and we're measuring KD here. So remember, lower KD is better binding. We also compared the MERS, SARS, and SARS-2 enzymes to the human macrodomain MDO2. What we found was that in all cases, MDO2 had better binding than the coronavirus macrodomains. But what was interesting is that these assays had different results if they were based on just binding to ADP-ribose, which is really the product of the reaction. It's not really the substrate per se. Versus those ADP-ribosylated peptides. And when you look at those ADP-ribosylated peptides, it's clear that the SARS-2 macrodomain seems to bind to those peptides better than the other viral macrodomains. We also tested, again, compared them in the hydrolyze assay, and at a ratio, we use different substrate to enzyme ratios here, 1:1, 5:1, and 10:1, so you could actually start to see multi-turnover events and how these enzymes behaved under those multi-turnover conditions. And at 1:1 ratio, all of the coronavirus macrodomains had similar activity, though they were significantly better than the human macrodomain. As we added more and more substrate, you see kind of that the SARS-2 macrodomain starts to distinguish itself over here and actually seems to have a greater initial velocity of that reaction early on. And so we conclude that the SARS-2 macrodomain binding to an ADP-ribosylated peptide and its initial enzyme velocity seems to be at least slightly better than the other coronavirus macrodomains. And I don't know if that actually matters in an infection or not. It does show there might be some interesting biochemistry here that's happening with these different enzymes. All right, so that's all I'm going to talk about with the enzyme activities. So we're now going to move on to talking about those PARP enzymes that actually mediate the ADP-ribosylation following an infection. All right, so we knew that these macrodomain deficient viruses were highly attenuated in vivo, and if you look at what was happening in vivo, we had reduced viral titers, and in some cases dramatically increased levels of cytokines such as interferon and CXCL10 and some other things. However, we didn't have a really great in vitro system for studying the role of this protein in cell culture, which is really needed to figure out what it's actually doing. So a few years ago, I decided to, using MHV as our model, we tested the ability of these deficient viruses to replicate in bone marrow-derived macrophage cells. And we chose those cells because, one, they're naturally infected by MHV. We know they play important roles in the disease caused by MHV. They have a very strong innate immune response, so we should be able to discern if there are those differences in those innate immune responses. And finally, we can culture a large number of these cells from an individual mouse and do fairly robust experiments. And so this is just a quick cartoon diagram of how we do this. We harvest the bones from the mouse legs. We harvest the cells from those, incubate them with a specific growth medium, and then analyze and infect on day 7. So remarkably, we found that in these cells, our macrodomain mutant virus was highly attenuated in terms of virus replication. And I want to point out this is a log scale here. So this is a greater than a log difference in virus replication, looking at platforming units or by a genomic RNA replication. And we found that in these cells, the mutant virus elicited a very strong interferon response compared to wild type virus. And that's looking at both mRNA and interferon protein. So our next question, I guess, the next obvious question was, is this due to PARP activity? Nobody had really ever addressed that specific question before. So to address that, we obtained some PARP inhibitors that are known to inhibit basically all PARPs. So they inhibit virtually all 17 human PARPs. And so we then looked at their virus replication. We wanted to ask whether or not adding these inhibitors would restore replication of our mutant virus and also diminish that interferon response. And so here you can see this is our virus. It's actually tagged with the GFP protein, and so you can see robust replication of our wild type virus in just DMSO treated cells, but our mutant virus, I don't even know if you can see it here, but there's very, very little green at all. Now, if we add these inhibitors, all of a sudden the mutant virus has dramatic increases in virus replication. There really wasn't any change in the wild type infection. And we have measured that by numerous methods, and we found that, yes, in fact, that those inhibitors don't really affect wild type, but specifically do enhance mutant virus replication. Furthermore, if you look at that interferon response, it is basically completely ablated when you add those inhibitors. So it does seem like, yes, PARPs are responsible for this attenuation. So next we looked for which, we want to find exactly which PARPs were involved here. And so this work was largely carried out by Matt Grunwald in the Perlman lab when I was there. He looked at the expression levels of all, actually 16 PARPs that are in the mouse genome. And what we found was that about half of them were highly upregulated upon infection, greater than 10-fold. And not surprisingly, these are the same PARPs that are interferon stimulated genes. And so we then took this subset of PARPs here that were upregulated following infection. We did an siRNA screen, and over months and months of work and probably dozens of experiments, we came to two major conclusions. First was that PARP-12, when you knock down PARP-12, and we used at least 2 siRNAs for every single PARP. PARP-12 was the only PARP that when you knock it down, we got substantial restoration of our mutant virus replication. So we had about a 6-fold increase here. With this specific siRNA, I think we looked at a number of different siRNAs, and they always substantially increased PARP-12 or the mutant virus replication, but had no impact on wild type, right? So as I got to KU, I decided I wanted to develop a better model for this. So we ultimately, with the help of Nancy Schwarting, my animal technician, and my first graduate student, Catherine Kerr, developed PARP-12 knockout cells. And Catherine then did the critical experiment here where we harvested those knockout cells and tested the replication of our mutant virus. And sorry, it's not labeled the same here, but it is the same virus. But you can see that in the PARP-12 knockout cells, we have virtually fully restored replication of our mutant virus, indicating that PARP-12 indeed is required for the antiviral ADP-ribosylation against our Macrodomain mutant virus. So what about that interferon response? And so I'm not going to show you the data from the actual siRNAs, but that data indicated that PARP-14 might be involved. And so we got some PARP-14 knockout cells from Masanori Ayakawa at the University of Harvard. And what we found was that in the absence of PARP-14, that increased interferon response you see here in our mutant virus infected cells is completely ablated in the lack of PARP-14. And then in collaboration with Chris Sullivan's lab at University of Texas, they developed a PARP-14 knockout human cell line and induced those cells with poly-IC. And you can see here that PARP-14 knockout cells had significantly reduced interferon production following poly-IC stimulation. So everything is pointing toward the fact that PARP-14 can enhance the interferon response, while PARP-12 is likely doing something to inhibit virus replication. Of course, we don't know that yet. We're actively trying to figure out what are the targets of these PARPs, and also are they targeted by the macrodomain. All right, so we don't think that that's the only thing happening here. And I'm just going to quickly point out some data from a undergrad and a technician in my lab, Lyndon Voth. And we found that in this BBT cell line, which is an astrocytoma cell line, these are astrocytes are infected by MHV in the brain as well, is that our mutant virus was significantly attenuated in these cells as well. But what was really interesting was that, when Catherine looked at the expression level of PARPs in these cells, there was really not a lot of PARPs that are highly expressed in these cells or significantly increased upon infection. And most notably, PARP-14 and PARP-12 are not there. So that indicates that additional PARPs likely have antiviral activity, and this may depend on cell type and even what virus they're responding to. All right, so that's what I'm going to talk about. That's the end of the PARP story, and obviously we're interested in looking at what's happening in SARS-2 infection. I'm just getting my BSL-3 about ready to go right now, so we're going to be actively looking at those questions, okay? But finally, I'm going to discuss the impact of NAD on this PARP macrodomain battle, and that substrate, or perhaps the weapon you could call it in this battle. So this all started with collaboration with Dr. Charlie Brenner, who, once SARS-2 emerged back in January and February, he was very interested in what maybe the virus was doing to the NAD during infection. Charlie has a long history, and he's been very successful in uncovering many of the enzymes that are involved in NAD biosynthesis. And so along with Colin here, they looked at the transcript data largely from Blanco Mello et al. From the Cell paper, and also from several other data sets and looked at the NAD metabolome, I guess, the transcriptome of proteins that can affect NAD levels here. So first I want to point out in CALU3 cells and A549-ACE2 cells, which are commonly used, cell culture models for SARS-2, that PARPs are highly upregulated in both cells. So this is very similar to what we see in MHV. But in addition, we found that a couple of different enzymes that are involved in NAD biosynthesis, including NAMT here, which is in both of these cell subs, are upregulated or even downregulated. So this NAD SYN1 was downregulated in a lot of different gene expression sets. So that all indicates that perhaps the NAD levels might be altered during infection. And so, as I said, I don't have BSL-3 yet here. So we used MHV infection to look at what the levels are of NAD were after infection. And this was done again by my postdoc, Youssef, and also Mark Schmidt at the University of Iowa, who's in Charlie's lab. And what we found was that after infection, and we did this with multiple cell types, that NAD was in fact dramatically diminished after infection and other NAD-related molecules. Then the Mike Cohen lab at the Oregon Health Sciences Center and Daniel Sanderson in his lab also showed that PARP overexpression can decrease NAD levels. So you can see, I'm just going to focus on these first and third lanes here. You can see that upon PARP10 overexpression, you get a significant reduction of NAD levels. So that leads us to the hypothesis that perhaps, during infection, that overexpression of those PARP enzymes is maybe diminishing NAD. And then we hypothesized that diminished NAD may actually affect the activity of these PARPs. And so what Mike's lab did was that he added a small molecule inhibitor to his cells, called SBI, that is an activator, sorry, not an inhibitor, but an activator of the NAMT enzyme that I just showed you was increased dramatically upon, not maybe not dramatically, but increased significantly upon SARS-2 infection. All right. So when he does that, he finds that the activity of these PARPs is always increased. So we looked at the overexpression of PARP7, 10, 12, 14, and even the natural expression of PARP1. And in every case, SBI significantly enhanced the activity of the PARPs, indicating that yes, diminishing NAD levels can maybe temper the activity of these PARPs. And as they're an important part of the innate immune response, it seems like enhancing NAD may be a good thing to counter the virus. Now, we hypothesized again that since our mutant virus that we talked about was in fact inhibited by PARP enzymes in those bone marrow cells, that perhaps increasing NAD levels would further diminish the virus, right? And so this is work, a great set of data set done by my student, again, undergrad, and then technician in the lab, Lyndon Voth, who's now actually a graduate student at the University of Missouri. And we actually tested 4 different precursors and compounds that can enhance NAD levels. So we looked at nicotinic acid, nicotinamide riboside, and nicotinamide itself, as well as that SBI molecule. And we used the cell culture model of MHV, where in these 17 clone 1 cells, our mutant virus actually is not attenuated. So the virus replicates perfectly normal on these cells. And perhaps that's because there's very little actual PARP activity.
Anthony, 2 minutes.
All right, yeah, I'm on the last slide. So what I thought was quite remarkable is that when we added each one of these compounds, we had diminished replication of our mutant virus, but virtually no effect on the wild type virus. And what you notice is that NA had the least effect, which is consistent with the fact that NAD SYN1 was somewhat downregulated upon infection, whereas all the other 3 had almost a log effect on the replication of our virus. And those enzymes over here, the NAMT and the NMNAT1 through 3, were more likely to be upregulated upon infection. All right. So this suggests that enhancing NAD through these, what we call salvage pathways, actually enhances PARP activity and inhibits a macrodomain deficient virus, not wild-type virus, in these 17 clone 1 cells. So the take-home messages from, I think, from this talk are that, 1, the SARS-2 macrodomain, as we expected, is an ADP-ribosyl hydrolase, and we have a number of assays to measure this by, and we're well equipped to kind of go after different mutations in this domain and identify their activities, as well as identify inhibitors of this protein. PARP-mediated ADP-ribosylation is a significant component of innate immunity. We've identified that 12 and 14 are involved in the response to coronaviruses, but there are likely others. And finally, that NAD is an important part of that PARP response. So it seems to be depleted by both PARP expression and virus infection, and that enhancing NAD can actually have an impact on virus infection. So, and final take home, I guess, is that the coronavirus macrodomain is a prominent virulence factor, potential therapeutic target for coronaviruses, and we're actively looking to identify inhibitors of this protein. And then finally, supplementation with these B3 vitamins, which are the NAD boosters, I guess, have the potential to enhance PARP activity and overall innate immunity and perhaps reduce virus replication. And we're actively looking at how that might affect in vivo infections. We think that you may be actually able to affect wild-type virus if we can get this NAD to cells that are pre getting infected. All right, so with that, I'd like to thank everybody that's been involved in this work again, and I'd be happy to take any questions.
Thank you, Anthony. Really very informative presentation. Let's see. You want to raise your hands for any questions? Okay, Rodney, you're up.
Very nice talk. Thanks so much. I just wanted to ask a question about macrodomain proteins in general. How many are there in human cells?
Yeah, humans actually have a number of macrodomains. I think it's like 9 or 10 of them. There's 2 of them that are in the same class as the coronavirus macrodomains. I think it's just MDO1 and MDO2. The others are somewhat divergent.
And part of the reason I was asking is PARP inhibitors have become important in certain cancers. And as you said at the beginning, PARP is important in stress response. And I just wondered whether you knew whether any of those proteins are involved in that stress response.
So none of the cellular macrodomains have been implicated in the cellular stress responses. I think they have been implicated in cancer. There's been a few papers, in the last few years. I think there was a PNAS paper that showed that some of these human macrodomains, if you delete them, they can have, I can't even remember the effects they had on cancer, quite honestly. It's been a while since I've read those papers. But there is a little bit of data out there on that. Not a lot, but there is starting to be more and more data on that.
All right, thank you.
So Anthony, so there have been recent results from other inhibitors that represent sort of oncology drugs, and some of them have been not disappointing in terms of controlling infection, but disappointing in terms of overall mortality. For instance, a recently conducted study on selinexor showed that while the actual, infection, titer levels, et cetera, but various parameters related to the infectivity of the virus were in fact reduced, the overall mortality was higher in patients treated with these inhibitors than in normal patients. And I assume this is due to the toxicity that is associated with some of these oncology compounds. So PARP inhibitors are not completely toxicity free, and they may also induce long-term consequences because they tend to accrue mutation in the DNA. So have you thought about how to think about bringing this to the clinic, or are there indications that this may be well tolerated?
Yeah, so well, one, I wouldn't necessarily use PARP inhibitors in, for a virus infection. These are proteins that are part of the immune response. They're trying to stop the virus, so I don't think you'd want to necessarily inhibit the PARPs. So like I said, we're trying to more identify inhibitors of the macrodomain and try to develop those. Unfortunately, there's no inhibitor has ever been developed for a macrodomain before. And so we're really starting from the ground up and trying to really find compounds that have any effect, and we're going to have to improve them and stuff like that. We recently did a computer screen, a computer, look to see if we could find previously approved drugs that might inhibit the macrodomain. We got about 20 or 30 hits from that computer modeling, and we're testing those to see if they might inhibit the macrodomain. But most of our results right now are pretty negative on that. So computer searches for those kind of compounds are always kind of tricky.
I think we have another question from Steve.
Yeah, hi, thanks. Beautiful stuff. So, I thought it was interesting you have macrodomains that hydrolyze and macrodomains that just bind. And maybe they're just binding and blocking accessed by somebody else. And I was thinking about it because of the context of PARPs, because there are all the PARPs that are active transfer guys, and then our favorite PARP is PARP 13, I think it is, that's inactive. So it's a domain mutant that only binds.
Yep.
So maybe there's, I don't know what, maybe there's competition between macrodomains and inactive PARPs as binders, and maybe just binding and covering is a thing.
Yeah, that's actually a really, I hadn't totally thought about that idea of it's preventing those PARP 13 or PARP 9. And actually a lot of PARPs even have their own macrodomains. So PARP 14 has macrodomains as well that bind to ADP-ribose. And it could very well be that. Now I have some data, I'll, I didn't present it here today, but we've made some binding mutants for the macrodomain. So very specific that, that would impact the binding of the macrodomain. And we have different phenotypes with that than we do with our hydrolyze deficient mutants. And so we're really interested in continuing to dive into that and see what PARPs might be involved. But I hadn't thought about the non-active PARPs, and I think that's a really unique, something we should look into.
Yeah, because maybe they're almost like macrodomains in the
almost.
Yeah.